Hi, this is Dr. Ben Morrill. Welcome to episode 83 of Reptile Genetics Weekly. Glad to have you here and excited to be able to talk about VPI Axanthic. Very cool to have a publication now where we be able to talk about some of the specifics, the gene and what causes it, what's happening, why we get an axanthic looking animal. So pretty cool when we get things like this. It doesn't happen every day, so. As for updates for samples, we have the fast test going out pretty much every day, sex determination going out usually twice a week. Those are still flowing well. And then the panel update, I can finally give you specifics. Once again, as long as the run goes well. The run is actually going right now as we're recording on Monday. So by the time you're watching this Tuesday night, I should, as long as the run goes well, I should have the results and be analyzing them. So I'll be analyzing Tuesday and Wednesday and be able to start posting them probably Thursday and Friday and the weekend, hopefully at the latest. So they're all those panels are coming out here pretty quick by the end of the week. Yeah, that's what I have for updates. Kayla, how are you doing?
Hey, Ben, I'm doing good. I'm excited to talk about VPI Axanthic today. It's been a while since we've done our monthly-ish morph spotlight, which is why we call it the monthly-ish morph spotlight. But it'll be cool. It's really neat to see like this actual science behind the genetics that we play with as keepers, and the artistry behind that. So really excited about it. But for let's do business stuff real quick. If you haven't heard already, we are trying to make sure everybody knows that there is a 25% discount on all shed tests on Morph Market for the month of November. So definitely take advantage of that while you can. You can buy a whole bunch of shed tests in advance, and you can use those throughout the year. So if you plan on having a bunch of babies this summer, you can buy the stuff now, get a steep discount. You can even email Sean at raregeninc@gmail.com, and he will, and if it's a big enough number of tests that you're planning on, we can possibly even give you a steeper discount than that. So shoot Sean an email, raregeninc@gmail.com, if you plan on doing ordering in bulk, or if you're just doing a few, you can still get a 25% discount this month on Morph Market. So don't miss out. It's awesome.
One other thing, I've had a few questions come through. People that want to do a 30 gene panel, they're not sure what they have, but they also think like it might be chocolate or it might be fire or something like that. So the newer tests that aren't part of our 30 gene panel. So you can do it through Clutch or Morph Market where you pay for the panel. And then if you just want to add one more, it's just like a $25 add-on. So you can do that pretty easily through either one. We haven't really mentioned that in the show, but that is set up so you can do that pretty slick and quick if you want to add one of the newer tests. And at some point in the next few months, we'll probably update our panels again, and we'll probably do a show about that and kind of talk about what will be the most helpful for people. But anyway, for now, you can add one of the new tests. Each one you add is like a $25 add on top of the panel.
Not too bad. For those of you who are looking to do who have been wanting to select morph tests, maybe not a full panel, but wanting to do like a la carte, I want to do this one, and maybe not pay the full price for each one because that does get kind of expensive. You can bundle them. They're bundles on Clutch, and they're called Build Your Own on Morph Market, depending on which one you prefer to use. Both of them are our partners. And so if you want to do a 2-test bundle, that is just $75, and 4 or more, for example, on Clutch, it's $110, and then just add $10 each for each additional mutation. So it's awesome. Definitely recommend it. It's the same price for both of those, both Clutch and Morph Market, for the a la carte style bundles. Bundles, build your own. There's like 3 different terms. They're all the same. They're great. Go check it out.
Yeah. And then the question might come up for the Morph Market 25% off. Is that, not count if you're doing the build your own? But it does. So whatever the discount you would normally get through Morph Market for doing a build your own, or like doing a small panel or a large panel or whatever, the 25% is an additional discount, and it'll automatically get added when you're checking out.
That's awesome. Stack them up, save some. And we went to a show this weekend, and by we I mean Sean. So we had the St. Louis, St. Louis, however the locals say it. Don't come for me. At the NARBC reptile show, and so that was this past weekend. We looked super official. We had our own table. Sean sent me this picture where we had all the banners, and we had our show exclusive shirts there and lots of stickers. So hope you guys definitely took advantage of that. And Sean sent pictures of some of the folks he met there, like Yeti Morphs. And I just now realized why I think this guy goes by Yeti Morphs. That is a very tall man.
Yeah, he is not petite.
No. Have you met him before?
Yes.
Yeah.
I think he's always at the Tinley show, but I think I've at least seen him at Tinley each time I've gone. Yeah, he's super nice, and I believe he's a Texan also.
Oh, okay, makes sense. And apparently he's a dog breeder too, because a couple years ago Sean got one of his pups. And I forget the breed that he said it was, but this is a younger picture of the pup that he got from him. And she's apparently like 50 plus pounds now. So really cute pup. And I mean, that's awesome. Get your snakes, get your pretty dogs. And that looks like such a good girl. So it's awesome. And TNA Chameleons was also there. Sean was talking with them about us possibly moving into locality testing, which I know is very, very sought after in the world of chameleons.
Yeah. We've made progress on it for panthers.
Ooh, that's exciting.
Yes.
Also, I love the setups they have there. Like, you can't see a lot of it in the pictures here, but that's honestly a really impressive setup, even at a show. Like, that looks really cool. I don't know if they had animals in there or if it was just a here we're showing off the kind of displays that we have for our animals. Either way, looks really nice, really professional. And that's awesome that Sean got to meet up with them. So recently, I heard from you that you got a shed from someone who sent in a shed from an all-white snake. And so, probably an ivory of some kind, but there was an interesting situation with it. So what's going on there?
Yeah. So this is another situation. First of all, it's an all-white snake. We had the episode a couple episodes back with Justin Thomas where we found out some cool results. All of the mutations his all-white snakes were hiding, and can use testing to figure out exactly what's going on.
Yeah.
In this case, it was an all-white snake, and this one as an ivory, and then this one could either be leopard or super leopard. It could be spot nose or super spot nose. It could be het clown, or it could be an actual visual clown. And so there's just lots of possibilities. Really,
and it's an all-white snake, so you're not going to see that otherwise.
Yeah. So the person purchased it kind of hoping it's just kind of a question mark plus, like, you don't know for sure what you're going to get.
Right.
This one was purchased far enough back. We didn't have tests for all of this, but raised that girl up and did send a shed in. And so it was pretty interesting, and also a little concerning what the results came out as. So the results said that it was a super leopard, and it was a super spot nose, and it was a clown.
All homozygous.
Yeah, and then obviously with the ivory, that's homozygous also. So the first thing I asked was, hey, is this a female? She's like, yeah, it is a female. I was like, this might be parthenogenesis. You have that many things that are homozygous. It's a little worrisome. So I said, how is this animal done with feeding and growing? And we kind of talked a little bit about a lot of the time partho animals are slow to grow, and if they do reach sexual maturity, usually they don't reproduce. She's like, oh, well, I just got a clutch of eggs from her.
Look at that.
Yeah, so she's got a super leopard, super spot nose, ivory clown female that does seem like it wasn't partho. It laid 8 great eggs for her. They all have veins and look good. And
wow.
So, yeah, I mean, that's talk about killing the odds when you purchase something that's possible for all those things, and you end up getting homos for everything that you could.
That's awesome. Good sized girl too, from the looks of it.
Yeah.
Oh,
and
that is
for a first clutch, 8 big eggs. That's really good.
Oh, that's awesome. Well, very cool. Good job hitting the odds, Okie Mom Balls, or K, from Okie Mom Balls. So that's awesome. And
congrats.
Congrats. Too cool. So this is actually related to a different story. So Ben, you a couple years ago, you sold one of your females to Stacy Carter, of Old Devil ball pythons. And recently something interesting popped out. So what's the story with that?
Yeah, so I mean, it was probably like 3 or 4 years ago. And this girl is all grown up now, and she got a clutch from her, and she ended up, Stacy bred an Ultramel male, Ultramel combo male to her, just to make some hets.
Yeah.
Some Ultramel hets. And so that's pretty cool. And this girl that I produced, the mom to her was an Odienchi, and that I hatched out the mom to this mom. So I guess the grandma to this clutch that we're about to look at. I hatched her out, and I bred that line several times. I know very well what's there and what's not there. But the sire to the clutch that this girl came from was Super OD, Enchi Spider, possible yellow belly, possible het clown that I got from Bob Vu, so Bob's Balls.
Yeah.
And so I hadn't bred very much, and I ended up, I can't remember, trading or selling, but I don't have him anymore.
Right.
But anyway, so one side of the pedigree I know very well. The other side I don't know for sure. But I just knew what Bob had told me. And then Stacy reached out this last week and showed me this picture of one of the babies that popped out.
Surprise!
Yeah, that thing is bright.
That is a glowing Ultramel.
Yeah, so the female was a Super Orange Dream, and apparently she was het for Ultramel, which I didn't know. So that meant from Bob, more than likely, I'll have to ask him when I talk to him again or see him at a show again and see if he remembers that male that was Pos Het Clown and Pos Yellow Belly, I guess, was also Pos Het Ultramel, and that proved out.
Yeah, it proved out, but it's only, 1 or 2 generations down, so that's really funny. But hey, that's awesome. Good looking clutch, and it just hit the odds.
Yeah.
So how cool is that? And congrats, Stacy. Those are good looking babies, and I hope everybody starts feeding easily and without too much hassle.
Yes.
Awesome.
Yeah, it's fun to see animals that I hatched out grow up and have babies of their own, and I love that kind of stuff.
Oh yeah, it's super satisfying. I know when I had my first clutch of rat snakes, Scott Hitchcock, who's the founder of the lightning gene in rat snakes, when I had my first lightning babies pop out, he messaged me on Facebook and was like, "Okay, so are these my great grandkids?" And I was like, "Yeah, pretty much."
Yeah.
So it really brings you closer to people, in a way that's different from a lot of other hobbies. So very special. So let's get into our monthly-ish more spotlight, and this time we're talking about VPI Axanthic and the technically pre-publication paper that just came out about it, with a few very familiar names.
Yeah, so this is from the Canadian group we've talked about many times. They're the first ones that came out with a ball python morph genetic analysis and paper. So this is Alan Garcia Elfring and Dr. Heather Roffey that we've talked about many times.
Very familiar names.
Yes, and in this case, they found out that the VPI Axanthic is caused by a gene called GTP
cyclohydrolase 2?
Yeah, cyclohydrolase 2. Or
say that 5 times fast.
Yeah, we'll say GCH2. That's a little easier.
GCH2 sounds good.
Yeah.
Awesome.
it's been long enough since I was in like OChem and stuff like that. I haven't had to say those chemical names in a while.
Oh, that's fair. Oh, I bet tongue twisters in chemistry class or like organic chemistry classes must be crazy.
Oh yeah, and some
uh,
very nerdy tattoos.
oh yeah, that's true. Yeah.
For sure.
All right. So tell us about what that means.
Yeah, so it's interesting the morphs we've talked to up to this point, and
Mhm.
kind of like in general for vertebrates when they have color and pattern differences. A lot of the time it has something to do with melanocytes or melanin, or what we talked about, like with hypo. The reason why they were a lighter color is because there was a problem with the transportation of the melanocytes, or the melanin was still being made. It just wasn't being transported out into the cells that you see when you're looking at the snake.
Yeah.
So it was a transport problem. In this case, what they learned was that for these axanthic animals, there was a certain type of pigment that the production of that pigment was hindered. So once again, it's different because it's a yellow pigment. It's not anything directly to do with melanin or melanocytes. In this case, it is a pigment, a yellow pigment, and so in the xanthophores have these yellow pigments, and they can be produced from pterins.
Mhm.
Starts with a P, so it's kind of a confusing one, but
It looks like pterins,
yeah,
but it's not.
pterin, like a
Pterodactyl.
pterodactyl.
Exactly.
So in this case, this GCH2,
Mhm.
when it is mutated, what happened, and I think on the next slide we have,
Mhm.
oh, so this is,
Yeah.
so we've talked about this kind of a slide before, but I don't know that we've shown it. So this is cool because you can see when we do the sequencing that we do when we're finding a new mutation, where we sequence, in this case, a bunch of individuals that are VPI axanthic animals,
Mhm.
or a bunch of, and then another group where they don't have VPI at all. And then we look for peaks. So if you look across there, you can see in that second chromosome, you can see as you go along, you see kind of, a little bit of signal, and then you see like a column going up, and then it has that GCH
Yeah.
one there. And so that column going up, that's what we're looking for to tell us, okay, that's likely the location on the chromosome where the mutation is that we care about. And then they went in, looked at GCH1 and GCH2, and found out, okay, it's GCH2 that makes the difference. So here, what this is showing for this GCH2, the mutation is just a single nucleotide polymorphism, or a SNP we've talked about before. So it's
Mhm.
just one base that changes. But in this case, this specific case, where that one base is, it actually causes a stop codon. So there's A's, T's, C's, and G's. There are three in a row. Those are called codons, and they code for different amino acids. But there
Yeah.
is code that says, okay, this protein's done. Stop. We're done. No more protein, and it gets clipped and it becomes a protein. Well, in this case, where that one SNP is, it actually changes it from coding for an amino acid. Instead, it changes and codes it to be a stop, but it's not
Huh.
where it's supposed to stop. So you
Interesting.
can see on the wild type on the left, you have all those coils going through, but the one on the right, where you have a VPI axanthic, what that protein would likely look like, and it's just truncated. It's shorter than, and
Huh.
apparently in the function in the cell, that actually changes. It makes it so that those pigments can't be made. The yellow pigments won't be made, and so
Oh,
without the yellow pigment being made, obviously can't show up in the skin, and we get an axanthic ball python.
hang on. I'm counting something real quick. I have to make a really silly joke. Give me a second. Uh, 6. Okay, we're gonna say that's like 30. Okay, so what you're saying is VPI Axanthic animals are like 30 30 not codons, 30 base pairs lighter than wild types. In weight. Because that totally works. Totally makes sense.
Their protein would be lighter, yes.
Amazing.
Yes.
I don't think you could weigh that out on a kitchen scale, but you can dream. But okay, cool. That actually is really neat. I okay, so somebody else may ask this too, but there's under the wild type, there is like right above my head actually. There's a little star. Does that mean anything that affects us?
That's
Yeah.
where the snip is.
Oh, okay. I was thinking it was a little green dot.
Yeah, I believe that's where the snip is. That green dot, I'm not sure what that is, but the star,
Okay.
I believe, is where it's at. Because you've got 3 loops at the top, and then you've got a break.
Mm-hmm.
And so that
Oh,
matches.
so that's the part that is cut off. Everything after the little star is the.
Yep.
Interesting. Okay.
Yeah, it could be that. Yeah. I'm pretty sure that's what it is.
Yeah. Okay. Very neat. So, and I guess in the graphic here where it has all the different colored ball pythons, I guess those are all the wild type, and then the rest are VPI Axanthic examples.
Yeah, and they kind of talk in the publication about different like melanocytes and xanthophores and melanophores and how things move. So like the tri-stripe example there, they're kind of talking about how the different types of pigments interact with each other and how they move, and then also the super cinnamon versus the albino super cinnamon in the bottom. So any of you that want to nerd out some, you can just type in that title that we gave you for this paper, and anyone can pull it up. You don't have to have like an academic license. The way they publish this, it's free to the public. So
yeah, and I'll have it linked in the description too, so you can check it out.
Yeah. So yeah, they do talk about some more nerdy detail of how these different pigments are made and how they interact, and kind of like 2 of them are repelled from each other, and that is likely what causes the tri-stripe, and that's how they feel like they're fairly confident what type of a mutation in gene is causing cinnamon, and why you end up with an albino super cinnamon, why it's white and doesn't have any yellow. And so it's kind of interesting, and it's as good as we can know with what we have right now. Really, the best ball python mutation, the best information we have to have confidence that we know it's the right mutation is pied, because they did CRISPR that in a lizard and cause that phenotype. So this is the best that we know right now given the information, but it does seem to line up with what we see in other animals for these specific genes and the pathways where the pigments are being produced and transported, all that kind of stuff. These papers that are coming out with information on ball python morphs, that's the best that we have right now, and seems very feasible because of what we know, like in mice and zebrafish and other things.
Right. And they were talking about in the abstract how basically like a lot of, as far as pigment genetics work, most of the work has been done on melanin and melanin production, whereas pterins that produce a lot of the yellow pigmentation have been understudied more or less. So hopefully this paves the way for more studies on that. So as far as testing goes, as we've discussed, it is a single SNP mutation that creates the VPI Axanthic mutation that we see and that we've identified as breeders. And so, Ben, how are we grading that one?
Yep. So this one is an A+, since we do have a publication. We've run thousands and other people, not only in the United States, but in Canada and and Europe. This is the same test that we're all using, and it's a very, very good test. Seems to line up when we run in panels. We'll run lots of samples in panels where people already know that it's either a visual VPI Axanthic or a het, and it's always lined up very, very well. It's very clean.
Awesome. And of course, as we've mentioned before, we give our tests an A if we've run over 1,000 known samples. But if it has a print publication to back it up or a pre-publication, then we give it an A+. So now it's time for our shed donor shout out. And I've got a little bit of a show and tell for this. So actually, let me bring this down. And Solid Serpents is donated sheds to us at Colubrid Fest this past year. Covering their phone number so we don't dox them, but I have a whole bunch of shed samples here, and I'm pretty sure it's mostly corn snakes that they sent to us. But we've got, what do we have here? Motley, Tessera. Oh yeah, we got corn snakes like crazy. But I know they have more than that, I believe. And I love that some of them have names actually written on them. Those make me happy when we have shed samples that have names. So, you have the Amel, Het Motley, but their name is Fluffy. I love it.
Awesome. Thank you so much.
Yeah, thank you. And Solid Serpents does have an Instagram. They have, I believe, a YouTube channel and Facebook account. And they work with more than just corn snakes. They have ball pythons. You can see a nice clown clutch there. Very shy babies. They've got some Pituophis. I think those are gopher snakes. And I see
candy corn.
Yeah, I like the candy corn a lot. That's awesome. And I think that's also a Ridley's rat snake down there too. So they've got all kinds of cool stuff. Colubrids, ball pythons, African house snakes as well. I know those were in there. I didn't get a picture of one, but they've got a great selection of animals. Good folks. It was great meeting them at Colubrid Fest. So thanks again. We appreciate you guys.
Definitely can't do it without y'all helping us, and we love seeing the sheds come in.
Definitely. All right, and I think that is all we have for today. So make sure you like, subscribe, give us a comment. It helps us in the algorithm, helps bring us to other reptile keepers in the hobby and other genetics nerds who feel like following along. So thank you guys, and I guess we'll call that a day.
Thanks so much.
Thanks, guys. Outro in 3, 2, 1.